Okay, anyways, 13 students I have recorded who all 13 students are present. Others will be marked absent for today. Please take a note of it, the people who will be joining later. Uh, so let's start with hybridoma technology. So basically, hybridoma technology is something that we use to make the monoclonal antibodies. And I had given a brief intro kind of yesterday in my lecture. So we'll start it again. Start with the history part and the principal part as well, just to revise what all we have learned in yesterday's lecture. And yesterday we almost finished like what do you mean by transgenic animals and how we can make transgenic animals. What is the process involved under in that? So, in 1975, this technology developed by George J. Kohler and Cesar Milston. So, and in 1984, they shared a Nobel Prize for this discovery. They make a hybrid cell. They made a hybrid cell that will make a number of monoclonal antibodies against an antigen. So that is what the hybridoma technology is all about. And basically, it deals with making up of a cell which is known as a hybrid cell. Okay. So the hybrid cell has the capacity of antibody production derived from the B cells of the spleen cells. And at the same time, it can divide continuously by the quality derived from the myeloma cells. So as we talked yesterday, we are going to fuse together the spleen cells and myeloma cells. So both of the once they fuse together and we get a hybrid, that will be called as hybridoma, and that will have a capacity to produce n number of antibodies against the antigen, that particular antigen that we have injected into the mouse. And also at the same time, it will be multiplying itself at a very faster rate continuously because of the property that it has got from the myeloma cells. So by combining the desired qualities of the both cells, the technology ensures large antibody production of the single specificity. The specific hybridomas, that is spleen cells and myeloma cells, obtain a monoclonal antibodies in artificial media. And this technology is called as hybridoma technology. So basically, production of monoclonal antibody with the help of this hybrids in artificial media will be called as hybridoma technology. Okay. So this is what we had learned yesterday, and very nice diagrammatic representation. So first of all, you will be taking a mice, you will be injecting that mice with the particular antigen, and then you will be uh, extracting the spleen cell who are going to produce the antibodies against the antigen that has been injected. Then we have this cancerous plasma cell, which is known as myeloma, and these two cells will be fusing together, right? And then we'll be getting a cell that is called as hybridoma, and that will produce n number of antibodies, and it will multiply also at a faster rate. Okay, so that is how the technology has been developed to develop. It's like people are making that in order to make the antibodies, monoclonal antibodies. See, currently, if you see from COVID-19 point of view as well, what must be happening? That people are trying to inject this antigen only, this coronavirus protein, enzyme, or some antigen. So virus for us, the virus is an antigen, right? So you will be inserting that antigen into the mouse, and then you will be checking if there are any antibodies that are being produced by um, the spleen of the mouse, and if we can increase the production of these antibodies with this particular kind of a technology or any other modified technology, then those monoclonal antibodies for that particular antigen can be developed, and those can be given to people for the treatment and also as a part of the vaccination. But generally, what we do is when we are vaccinating somebody, that means we are trying to make sure that the immune system of the body is going to produce those antibodies which can tackle that particular antigen or virus, right? So Either you can do that, or you can treat the people who have already gotten this disease with the antibodies, right? So people are also using plasma technology, in which they are using the antibodies from the people, the plasma of the people who have already got this disease, and their immune system has have not, the B cells. The B cells are the one, uh, B cells and the T cells are there in the immune system. So B cells are generally the one who can memorize each and every antigen, right? So अगर next time वही antigen वापस से body में होता है, तो B cells have the capacity, with the help of bone marrow, to produce the antibodies against that antigen, because they have the memory behind their back, remembering that particular antigen. That's the reason people are saying that, means like the scientists say, according to current research of COVID-19 disease, that if a person is infected with coronavirus, maybe for 3 months, okay, that person might not get disease again. He might not contract the disease again, because with the help of this B cells who can memorize the antigen who had infected their body before. But that is so that depends on completely on our immune system, whether they can memorize for lifelong or whether they can memorize for a certain period of time. So we are still investigating that how long our B cells can memorize this particular antigen, and how rapidly the production of antibodies will be happening inside the body, right? So that is how the whole technology, like that is how things work. Okay, so our immune system plays a major role for tackling any kind of antigen that we are uh, going to get contracted with. So over here also, this is artificial production of monoclonal antibodies, and whatever happens inside our body will be like a natural response to any kind of antigen that is going to come inside our body. So, in case of COVID-19, people are saying that if we can memorize the antibodies at least for 3 months, that means our body can, means bone marrow can increase the production of antibodies, even if in those 3 months the antigen is again entering into our body. And uh, if I simplify this procedure, and if I tell you that we have B cells who can memorize the antigen once the disease has happened to us, and then with the help of bone marrow, say the bone marrow is a place where faster production of the antibodies will be happening. Okay, so next time when the antigen is entering, at least that time, our body will recognize, yeah, this is the same antigen who had come before. So let's start making antibodies that we had made prior to tackle this particular antigen. And this time the production of antibodies will be much higher than the first one. Because in first contraction of the disease, our body must have taken a very longer period of time to understand what kind of antigen this is. And then once they start making the antibodies, by that time patient might suffer for a long duration. So for the second time contraction, maybe our body can recognize, and we can produce more antibodies than that of we contracted the disease very at the very first time. So that is how the thing is, and that is how the molecular level of understanding of disease. See, whatever we are learning right now, we can really relate to our COVID-19 pandemic situation, and each of the thing is applicable. I would say each of the thing is applicable, starting from your clinical research, starting from your immunology, starting from your this thing, what you say, restriction enzymes, your DNA sequencing, everything we can apply to this disease of COVID-19. So you can, means like you can really give your mind a little bit of a work and think in this area, understanding. So next part is that monoclonal antibodies. So what are monoclonal antibodies? So monoclonal antibodies are antibodies that are identical because they are produced by one type of immune cell only, and all clones of the single parent cell. Okay, and basically produced by white blood cells, which is called as plasma cell. Okay, just give me 1 minute here. Yes. So it is used for treatment of cancerous cells and anti-venom, that is anti-snake venom. So these are the monoclonal antibodies who will be very specific for one particular antigen only, and they are called monoclonal because they are produced by one type of immune cell and all clones of the single parent only. They are the clones of the single parent only. Okay. So that is the reason. So procedure for making the monoclonal antibody is they have jotted down in the 6 steps. First is immunization of the specific animal which generate the hybridoma cells with the spleen cells. So that is the very first procedure. Then isolation of myeloma cells. Then fusion between the spleen cells and the myeloma cells. Then selection with the help of hatch medium. It's a very selective medium, and that we have made to select only those cells which are hybrids basically. Then isolation of hybridoma cells and screening for hybridoma cells. So that is what the 6 steps are there in order to that we have to conduct in order to produce the monoclonal antibody. So the very first step is immunization of specific antigen. Okay, so an antigen immunized to an animal like mice via intravenously direct to the blood by injection, directly to the blood. You have to give it IV only. Okay, where in spleen it activates the B cells which produce the plasma cell that is called as spleen cell, then plasma cells to produce the monoclonal antibodies. Then we will go ahead and isolate these plasma cells from the spleen of the animal. So basically, once the antigen is been given, right, the plasma cells will be produced, which will be very specific. Which will be very specific in the production of the monoclonal antibody against that particular antigen only. Okay, so this is the procedure for immunization of specific antigen. Okay, so you'll be injecting the mice with the intravenous injection. Okay, then where in the after that, in the spleen, the B cells will be activated, and that they will start producing plasma cells. Those are known as spleen cells. Then these plasma cells will produce monoclonal antibodies, which are very specific to the antigen that you have injected into the mice intravenously. Then isolation of these plasma cells from the spleen of the animal. So we'll be taking this plasma cells and we'll be fusing this plasma cells with the myeloma cells. Okay. Then isolation of myeloma cells. So myeloma cells are cancerous cells which is isolated from bone marrow. So myeloma cells are generally immortal in nature, that is that which they never dies and has multiplication property. Okay, so we are going to isolate these myeloma cells from the bone marrow. Then fusion of the spleen cell and the myeloma cells. So first of all, it requires polyethylene glycol media for fusion. Okay, it can also done by electrofusion. Fusion between the spleen cell and myeloma cell cell produced 5 different type of cells. Okay, so we know that not all the cells are going to get fused together. We will have many different kind of products when we are trying to fuse this spleen cells with the myeloma cells. So we can get fused plasma cells, means plasma cells together fuse. Okay, fused myeloma cells, means myeloma got fused together. Then we'll get hybridomas, then we'll get unfused plasma, and then we'll get unfused myeloma. So these are the 5 possibilities of the cells with that we'll be getting once we try to fuse the spleen cells with the myeloma cells. You guys are understanding till here or not? Just typing yes or no. Okay. So we'll be getting 5 different type of cells, and what we have to do, you all know that we have to select only these hybridomas. So this is something we want, not this, not this, and not this. So this is something we want, hybridoma only. Okay, so now You have to do the selection of this cells only. How will you go ahead and do that? See, in the plasmids, when we were learning, that time we know that we are inserting a gene which can act as a selectable marker, right? That means we will be inserting a gene which will be antibiotic resistant, and then you can easily get only those plasmids which have inculcated our gene of interest in them. So over here, maybe more or less same kind of technology has been used. What do we have over here is, so selection of hat. So selection of hat, in the sense you will be selecting only, just give me 1 minute. This is kindly kind of used. Just give me 1 minute here. I'll just pause my share. Okay. So we have this particular media over here, and with which we are going to select our hybridomas only. Okay. So we have selection of hat medium, that is hypoxanthine, aminopterin, thymidine. Okay. So before multiplication of antibody, it has to synthesize. Just a minute. The content has gone now. Just give me a minute. So, yes, we can start. So, before multiplication of antibody, it has to synthesize new copy of DNA, and for that it requires synthesis of nucleotide. And for the synthesis of nucleotide, mainly two pathways are used. First is salvage pathway, and another one is de-novo synthesis. So in one salvage pathway, it requires degraded part of the old nucleotide to produce the new nucleotide, and in two, it means in de novo, it synthesizes completely new nucleotide by small molecule, that is sugar and amino acid. So in the selection of hat media, means like in the hat media, the myeloma cells, that is sorry, your hybrid cells, that is hybridoma cells, will have to undergo the making another copy of themselves before they can go ahead and produce the monoclonal antibodies. So uh, uh, they can do that with the help of two pathways. First is your salvage pathway, and another one is your de novo pathway. So salvage pathway requires the degraded part of the old nucleotide to produce a new nucleotide. Another one, de novo, is like it will completely produce a new nucleotide by a small molecule such as sugar and amino acids. So in Hath medium, cells not synthesized by de-novo synthesis due to presence of Aminopterin in Hath medium, which blocks the dihydrofolate enzyme which is necessary for this synthesis. So for synthesis in salvage pathway, it must require HGPRT enzyme, that is hypoxanthine guanine phospho-ribosyl transferase, whereas hypoxanthine and thymidine are used as precursors. So the cells are not synthesized by de-novo synthesis due to presence of aminopterin. So in hatch medium, this second pathway will not be available for multiplication because um, due to cell not synthesized by the novosynthesis due to presence of aminopterin in hatch medium, which blocks the dihydrofolate enzyme. So they require dihydrofolate enzyme to produce the cells, means multiply the cells by DNA replication will happen through de novo synthesis. For the de novo synthesis, we require dihydrofolate enzyme. But since we have aminopterin in the hatch media, this particular pathway will be blocked. Okay, so our DNA will not be multiplying. The hybridoma DNA will not be multiplying, या फिर replicating, I mean to say, through de novo synthesis. So the only pathway that remains for them to multiply their DNA is the salvage pathway. So for the synthesis in the salvage pathway, it must require the HGPRT enzyme, that is hypoxanthine guanine phospho-ribosyl transferase enzyme, whereas hypoxanthine and thymidine are used as the precursors. So we can use this pathway to multiply the DNA, या फिर replicate the DNA with the help of hypoxanthine and thymidine as the precursor for the production of the replication of the DNA with the help of a salvage pathway. So that is what they have shown. The spleen cells have HGPRT enzyme, isolation of hybridoma cells. Myeloma cells do not have HGPRT enzyme. So this is the interesting part over here. So, uh, so now we know that only those cells will be surviving who will be having the HGPRT enzyme, and they will not be able to. Another cells will not be able to multiply themselves. Okay. So the cells who can take up the salvage pathway and who have this HGPRT enzyme are the only one who are going to survive this hatch medium, right? And we also have added the precursor, that is your what was the precursor, the hypoxanthine and the thymidine. Okay, so spleen cell have HGPRT enzyme. We all know that the spleen cells already have HGPRT enzyme in them, but myeloma cells are the one who do not have the HGPRT enzyme in them. So fused plasma cell, in that the HGPRT is present. Fused myeloma cells, in that the HGPRT is absent. So obviously they are not going to multiply. In hybridoma cells, HGPRT is absent. In unfused plasma cells, it is present. In unfused myeloma cells, it is absent. So sorry. I'm very sorry. In hybridoma cells, the HGPRT enzyme is going to be present because it is fused myeloma and fused spleen cells. Since we have half of the spleen cell with us in hybridoma, obviously they are going to have the HGPRT enzyme with them, right? So spleen cells HGPRT positive, antibody producing, and myeloma cells HGPRT. So unfused spleen cell, they will die. Fused spleen cells, they will also die. Uh, and because they cannot multiply, right? You have to understand this part. Let me explain it to you again. So we have now we know that since we are going to take up the since cells are going to take up the salvage pathway to multiply the DNA, it has to have what? HGPRT enzyme. It is not going to multiply. The DNA is not going to multiply through de-novo synthesis because hatch media blocks the pathway due to as since it blocks the dihydrofolate enzyme. So de novo pathway is not of any use. So we need DNA to be multiplied with the help of salvage pathway. Am I clear till this point? Are we clear till this point? Okay. So after that, we in this chart they are telling us in that 5 combinations of the cells that we are going to get when we are trying to fuse the cells together, who all are going to have the HGPRT enzyme. So we know that fused plasma cells are going to have the HGPRT, and hybridoma are the one who are going to have the HGPRT. Then unfused plasma cells and unfused myeloma cells will not have. Infused plasma cells will have HGPRT, and unfused myeloma cells will not have HGPRT. That we all, because only the spleen cells are the one who are going to have the HGPRT. So we know that our hybridoma of our interest is hybridoma cells, and they are the one who are going to have the HGPRT, right? So these are the 5 combinations of the cells that we are going to get. So these spleen cells who are HGPRT positive, and these myeloma cells are HGPRT negative. So fusion in polyethylene glycol. Okay, so you will be fusing them in the polyethylene glycol medium, and then you have unfused spleen cells and fused spleen cells. They will die ultimately after one multiplication, after two multiplication. They will die because they are not like cancer cells who can keep on dividing themselves. Okay, but hybridoma are the one who will keep on proliferating because they have what? HGPRT, right? So they will be synthesizing their DNA with the help of a salvage pathway, and they will be multiplying themselves, and they will be proliferating. Okay? And whereas the myeloma cells will not survive in the hatch medium specifically because they are not able to use the de-novo synthesis pathway to multiply their DNA. We are asking them to use the salvage pathway, but can they use salvage pathway? No, because they are lacking HGPRT enzyme, right? So this way we are eliminating these cells. Just a minute, let me annotate. So this way, by using hatch medium, we are eliminating these cells, these cells, and these cells. The only thing that will survive is your hybridoma, right? So you guys understood how exactly the hatch medium is going to work on only specific selection of hybridomas only. So we can have the hybridoma which will proliferate. Okay. See, uh, spleen cells are the one who will eventually die. Okay. After one multiplication, two multiplications, maximum three multiplication, they will die because are not they are not someone who will survive forever like myeloma cells. But myeloma cells are not able to multiply themselves in the HAT medium because they lack the HGPRT enzyme. Okay? And then, and hence, the only thing that is surviving is the hybridoma, which is having half part of the spleen cells and half part of the myeloma cells. Okay? Are we clear to here? Have you guys understood this part? Okay. So, fused myeloma and unfused myeloma didn't have HGPRT enzyme, so can't survive in HAT media. Fused plasma and unfused plasma have HGPRT enzyme, but they don't have a long life, so they will die ultimately. So, hybrid cells has HGPRT enzyme from spleen cell as well as they have the ability to multiply repeatedly as myeloma cells. So, isolation of hybrid cell because it's only cell which survive in the HAT media. So next part will be screening of hybridoma. So ELISA screening method which done by incubating hybridoma culture in which secondary enzyme gets conjugated and formation of the color product shows the positive hybridoma. So simple ELISA technique will be used which will be using enzyme substrate reaction method and change of color which will confirm the production positive hybridomas. and used for multiplying the hybridoma cells in vivo or with the in vitro method. So in vitro will be like you will be doing it in the lab. You are asking, will you be asking, means like you will be making sure that hybridoma cells are multiplying with the help of a media. And in vivo, you will be injecting this hybridoma cells back into the spleen cells of the mouse and waiting for the antibodies to be produced by those hybridoma cells. So lots and lots. So in vivo procedure involves introduction of hybridoma cells into the peritoneal cavity of the animal. Then from ascetic fluid, antibodies are isolated. In vitro method involves culturing of hybridoma cells in suitable cultural media, and then antibodies are isolated and purified. So once a hybridoma colony is established, it will continually grow in cultural media like RPMI 16406. This is one of the cell line who will continue. This is one of the cell line who divides continuously and produce antibodies. And we can store these cells, hybridoma cells, in liquid nitrogen, which is maintained at -20 degree or lesser than that. Okay? Or minus 80 degree, in fact, I mean to say, or lower than that. So this is a very nice diagrammatic representation of hybridoma technology altogether they have given an application. So we can go through the application part quickly. I hope you guys, I don't want, miss, you don't need me to go through this diagrammatic representation again, but I would want you guys to go ahead and uh, read it yourself. Okay, so this is going to tell you about the same thing that we have learned by far. Am I clear till here how this hybridoma technology is working in production of monoclonal antibodies? Then we will go to the application part. Yes, I want to know from
Yes, ma'am.
chemistry people specifically. Who said yes, ma'am? Are the chemistry people attending the lectures or not, or they're already scared? Yes, Razina, are you getting it, Samruddhi? Hmm. Okay. Okay, okay, Shraddha. So we move to the application part for toxic. For serological application will be in identification of A, B, O blood group, diagnosis, detection of pregnancy by assaying of hormones with the monoclonal antibodies, separation of one substance from the mixture of a very similar molecule. So you can use specific antibodies for separation of few mixtures from the mixture of one substrate from the mixture of many substrates. Then immunopurification, purification of individual interferon using monoclonal. Inactivation of T lymphocytes responsible for rejection of organ transplants. In therapy, removal of tumor cells from bone marrow, treatment of acute renal failure, treatment of malignant leukemia, beta cell lymphoma, and a variety of allograft rejection after the transplantation. So these are the main applications of hybridoma technologies that we use in current, and the most common ones are used in for the diagnostic test. Okay, the most common one, the one we know, are mostly used in diagnostic laboratories. So this is what it is about hybridoma technology. Very simple technology to understand how. So, but you imagine how much of research people must have done in order to come up with such a protocol, which will help you in creation of monoclonal antibodies. And we all know that it's the need of the time. It's the need of the science and technology to have this technology been implemented. So it does not stop here. So you have to do the scale up of this thing. Whatever we have seen, you guys must have imagined it at the lab level or maximum at the until the mice study. But you have to scale up this uh, particular procedures. When I'm saying scale up in the sense that you have to produce it in the bigger sizes, you have to produce this hybridoma lot in lot many numbers, and you have to produce lots and lots of monoclonal antibodies so that you can dispense them into the bottles, and then you can dispatch them to the people who are in need. right? It's same as that of how we must be imagining the production of the tablets, the capsules. Same way you have to scale up the production of this monoclonal antibodies. So that is also a further part to it. If I find any information on that, I'll discuss in the next lecture how exactly the scale up is done. Okay, so we are done till here with the hybridoma technology, and we are almost done with the time. From tomorrow, we are starting the Google Classroom. Okay, so from tomorrow, we'll be meeting through Google Meet only, and so that we don't have to be restricted to the time constraints and etc. So we'll be sending you the timetable with the links that are you are supposed to join in with. Okay. So I'm signing off for now. If there are any doubts, you can ask me. We have less than a minute remaining. Are we clear till here? Okay. Okay. Then I'll see you guys at 1:30. Bye. Wait, Maggie. Maggie!